Welcome back to another YouTube video. It's your tutor, Disha. Today, I'll be working the Cape Biology Unit 1, June 2021, Paper 2. I've already done question 1, so today I'll be working the rest of the paper. Let's get right into it. Module 2, Genetics, Variation, and Natural Selection. DNA and RNA are made up of polynucleotide strands. Figure 2 represents part of a DNA molecule. Let's take a minute to look at Figure 2. Part 1. On the diagram in Figure 2, provide labels to identify the parts of the DNA molecule labeled S, T, U, and V. First thing you should ask yourself here is, what is DNA? DNA, deoxyribonucleic acid, right? It is a molecule that consists of 2 strands, and each strand has a backbone made up of a sugar and phosphate groups attached to bases. So now that we know that, we can go ahead and attach the names of the parts of the DNA molecule. S here at the 5' end is the phosphate group. T is a 5-carbon sugar, a pentose, and the name of this pentose is deoxyribose. Deoxyribose is linked to this base here. It is a purine called guanine. Guanine is also linked to another base here, a pyrimidine base, via 3 hydrogen bonds. According to Chargaff's rule, guanine always pairs with cytosine. So therefore, U is cytosine. Below cytosine, we have another purine here. It's called adenine. Adenine is also linked to another base here via 2 hydrogen bonds. And according to Chargaff's rule, he said that adenine always pairs with thymine. So there you have it, the labeled structure of the DNA molecule. Part 2, on the diagram in Figure 1, draw a circle around a section of the diagram to identify a nucleotide. What is a nucleotide, students? I hope you're not confusing a nucleotide with a nucleoside. A nucleotide is made up of 3 things. Yes, a 5-carbon sugar, in this case, deoxyribose, a nitrogenous base, and a phosphate group. 3 things. But the nucleoside, on the other hand, consists of a 5-carbon sugar, deoxyribose, bonded to a nitrogenous base. So since we know this, we can therefore go ahead and draw the circle around a section of the nucleotide. Part 3, state how 2 nucleotides in a DNA molecule are held together. Students, I want you to be extremely careful with what the question is asking for. Individual nucleotides are held together via covalent bonds, right? But polynucleotide strands, the 2 actual strands of alternated sugar and phosphate molecules, they are held together via hydrogen bonds. Nucleotides are joined together using covalent bonds, right? Moving on to part 4, DNA codes for polypeptides in cells. Different types of RNA are involved in this process. Outline the roles of messenger RNA and transfer RNA in the production of polypeptides in eukaryotes. First thing you should be asking yourselves, what is RNA? RNA, abbreviated for ribonucleic acid, is a single-strand nucleic acid. There are 3 types of RNA. There's messenger RNA, ribosomal RNA, and transfer RNA. So here the question says, DNA codes for polypeptides in cells. But here's the problem. DNA cannot come out of the nucleus. It's too large. So what happens is that the codes for making polypeptides, they're going to be transcribed in a transcript during what is called transcription, right? That transcript can exit the nucleus to the site of translation. So therefore, messenger RNA is that molecule that carries the coding sequence for protein synthesis. Now, when the messenger RNA with the transcript arrives at the site of translation, what will happen is that the transfer RNA will transfer the amino acids that match the mRNA codon to form a new polypeptide strand. Part 5. Sickle cell anemia is a disease caused by a change in the gene that encodes for hemoglobin. Name the chain in the gene which results in the production of abnormal hemoglobin in sickle cell sufferers. So the first thing that should come to your minds is gene mutation. And recall, gene mutation is a change in one or more genes. And in the case of sickle cell anemia, what happens is that there is a change in the gene due to a substitution mutation in the 6 triplet of the gene for beta globin. Part 6, the hemoglobin that is produced in sickle cell anemia leads to sickle-shaped red blood cells under low oxygen saturation. Outline how low oxygen saturation results in a change of the shape of the red blood cells. So in low oxygen saturation, what will happen is that the hemoglobin chains will form a hydrophobic contact between its amino acid valine on one chain and phenylalanine on the other chain, which will distort the shape of the red blood cell. Moving on, in 2017, French scientists announced that gene therapy had been used to successfully treat patients with sickle cell anemia. Figure 3 shows that results which detail the patient's results to low oxygen levels after 6 months of treatment using gene therapy, as well as the response of 5 control subjects to low oxygen levels. And my advice to students who are attacking the graphical questions, first thing you need to do is ensure that you read the questions carefully. You read the graph title, that can give you clues to the answer. You spend a minute to interpret the graph. And the corresponding question says, analyze the success of gene therapy's treatment for sickle cell anemia. What I would do is I would discuss the success of the patient after 6 months versus the patient after 12 months. This could indicate that gene therapy was more favorable over longer periods of time. This graph also required students to discuss 3 challenges which may limit the success of gene therapy as an effective treatment for diseases in modern medicine. So my advice to students is to let your discussions be evidence-based, right? Recall in class, there were 2 major experiments. Recall the one with the severe combined immunodeficiency syndrome, SCID, and cystic fibrosis. You could link those evidence to the limitation that arise from them, right? So you could talk about unwanted immune system reactions, in that when genes and vectors are introduced, they can initiate an unwanted immune response. You could talk about the possibility of targeting wrong cells. For the severe combined immunodeficiency syndrome, SCID, recall that patients develop leukemia due to uncontrolled cell division. So there's a possibility of causing a tumor here. could link the high cost of developing a treatment in clinical trials, or you could link the fact that most patients can't afford gene therapy here, and this may limit its success. In 3, data on the relative levels of hormones FSH and LH in the blood during the menstrual cycle are provided in Table 1. Table 1, relative levels of hormones during the menstrual cycle. All right, guys, the first thing you should be doing here is taking a minute to examine the table. And the corresponding question here says, on the grid provided, plot the data in Table 1 as a line graph with the day of the cycle on the x-axis. And I used my Excel app to plot a beautiful graph here. Always remember your title. That's a whole 1 mark there, right? So you put an appropriate title, Figure 1, the levels of hormones during the menstrual cycle. Ensure that you include your key here to indicate which hormone is FSH and which hormone is LH. And recall, they ask you for day of cycle on the x-axis. Here, your title of the axis here. So you'd have day of the cycle here versus the hormone concentration in IU on the y-axis. Part 2, outline the roles of the hormone LH and FSH in the maturation and release of the secondary oocyte during the menstrual cycle. First things first, what is LH and FSH? So these two hormones, luteinizing and follicle stimulating hormones, they are secreted under the control of gonadotrophin releasing hormone from the pituitary. And these have a direct effect on the gonads. The next thing you should be considering is, what is the menstrual cycle? Recall, the menstrual cycle is a 28 days series of events controlled by hormones. Recall, there are 4 main phases of the menstrual cycle, and these include menstruation, follicular phase, ovulation, and luteal phase. And since they ask about the 2 hormones, luteinizing and follicle stimulating, then we will focus on the follicular phase and the luteal phase. What this hormone specifically does is it stimulates the growth of ovarian follicles in the ovary, right? Or what we would say primary oocytes. And this occurs prior to ovulation. Now, during the luteal phase, there will be a surge in luteinizing hormone, right? And this surge causes the ovarian follicles to rupture and release a mature oocyte, or what we would say a secondary oocyte. And this is called ovulation. Now, the remnant of the ruptured ovarian follicle is called the corpus luteum. Luteinizing hormone therefore stimulates the corpus luteum to produce progesterone and estrogen also. Estrogen is going to build the lining of the endometrium, but progesterone is going to maintain it. And both estrogen and progesterone are required to support the early stages of pregnancy if fertilization occurs. Now, Part B, following the release of the secondary oocyte, fertilization normally occurs in the oviduct and implantation of the blastocyst occurs in the uterus. Outline the process of implantation of the blastocyst in the uterus, including hormonal influences. All right, the first thing you should be thinking about is, what is a blastocyst? The sperm fertilizes the egg. It will form a zygote. Following zygote formation, there's going to be a series of cell division. The first structure that's going to be formed after cell division is called the morula. The morula will continue to divide to form a hollow, thin-walled structure called the blastocyst. The blastocyst has 2 parts. It has an inner layer of cells and it has an outer layer of cells. Now, the inner layer will move on to form the embryo, while the outer cells, what they do is they will burrow into the wall of the uterus and develop into the placenta. And recall, the placenta is that organ that forms an interface between the mother and the fetus. Since they ask for hormonal influences, the placenta is special in that it's going to produce its own hormone. It's going to start to produce human chorionic gonadotropin hormone. Part C, in plants, both sexual and asexual reproduction can occur. In sexual reproduction, floral structures develop into a mature fruit following fertilization. A diagram illustrating a longitudinal section through the ovary of a flower is given in figure 4. So the first thing that you should do is take a minute to examine the figure here and see if you can identify some parts. So since this is the female anatomy of the flower, we would therefore say that here is the stigma. This is the style where the pollen tube will grow down. Right? This entire structure here is the ovary, right? Sitting in the ovary, we have the integument here. At the start of B is called the nucellus. Inside the nucellus, we have the polar nuclei here. There's one here, there's one here, and there's also a synergid. Okay? And the corresponding question says, state the change in each of the labeled structure A and B in relation to the development from floral to fruit tissues. A will develop into the fruit. Integuments will form the seed coat or testa, and then B overall will form the seed. Last question, discuss 3 advantages of asexual plant reproduction for agriculture. Include 1 limitation for each of the advantages discussed. So the first thing you should be thinking about is, what is asexual reproduction? Asexual reproduction is the production of a new offspring using one parent. And the first thing that comes to mind here is micropropagation. Micropropagation is one of the easiest ways to propagate seedless plants in agriculture. Scientists can extract tissues from plants and they can grow them in culture to generate many plantlets. And the good thing here is that they can change the genotype and the phenotype of the plantlets to resist diseases and so forth. But a limitation to this is that this process has to be done under aseptic conditions, which, is not always available. And what is asexual plant reproduction without vegetative propagation? The advantage of this is that through vegetative propagation, the farmers can propagate identical qualities as the parent plants. But a disadvantage to this is that there is no opportunity for genetic variation. Asexual plant reproduction is very important in the field of horticulture. And in horticulture, farmers, they use techniques like grafting, cutting, and layering. For example, grafting is very advantageous in the citrus industry, in that scions capable of producing a particular fruit variety are grafted onto rootstock with specific resistance to diseases. However, the limitation to this is that you can only graft a plant that is of similar species. All right, guys, that's it for this paper. Thanks so much for watching. Now you see the type of work that you have to put in here to get a grade 1 in biology. It's not a walkover. This is not CSEC biology. This is a big step up, right? It requires more reading, more practicing. Once again, thank you. Please subscribe to my channel and share with your peers, and be sure to attend my biology webinars. Bye.